Many thanks to the organizers for inviting me to give a talk on this conference. And I would never imagine it when I was listening to the lecture of Alexander Ivanovich Oparin at my first day in Moscow University. Lena remembers a lecture about nutrition growth and coarser weights, swimming there. But my lecture will be about microorganisms which are very well adapted to extremely harsh environment and no nutrition growth at all around them, quite the contrary. And of course it's a great honor and great challenge also to give a talk after these two previous brilliant lectures, but also a piece of luck because you got all the basic things and I now can not to stop them too long. So this is the summary of my talk. Few words about Archean biosphere and about its modern analogues, thermochemicals, and next 2 points they make the whole talk. The metabolic diversity of thermophilic prokaryotes, and especially the diversity of the catabolic reactions, the electron donors and acceptors. And in the end, a few words about new metabolic groups and how we can detect them in thermal environments, and about carbon cycles in general in these environments. But before start, also I would like to acknowledge my teacher, Georgy Alexandrovich Zavazin, who passed away 3 years ago. He was always very interested in evolution of biosphere, and he loved unusual microorganisms. He started working in thermal environments in the end of 70s, almost at the same time as Thomas Bloch and Karl Stetter and Wolfgang Zillig. So we were working in parallel, and Georgy Alexandrovich had a lot of new ideas, and we still continue using them. So Archean biosphere, just very briefly, because we already heard a lot. The dates, approximate dates, are for billions years ago and up to 1.5 billion years ago, and the surface of Earth was hot. There are different figures. I found this, which suits me best of all, and that's why I'm presenting them. And of course, it was anaerobic, and everything was reduced. And we now find the same conditions in different types of thermal environments. These terrestrial hot springs, this is Kamchatka and shallow water hot vents, and Kuril Islands, and deep sea hot vents. You have to do submersible to get there, and helicopter to get to Kamchatka, so it's not so easy to get there. And why this oil well is here, you know now from the lecture of Professor Stetter, because in deep subsurface we also have hot environment with temperatures appropriate for the growth of thermophiles and hyperthermophiles, but I will not touch this subject now, and let us go to terrestrial and submarine environments, hot environments, and here is another version of the tree of life, and I've marked hyperthermophiles with red, and moderate and extreme thermophiles with yellow, and brick lines are the branches where there are both mesophiles and extreme thermophiles, and straight lines are where there are only thermophiles. And you see that thermophiles are distributed through the whole tree of life, and the deepest lineages, as you also have seen from the tree Professor Stetter showed now, they are thermophilic. But what about the metabolic types? And especially metabolic types which could survive in the environments of Archean biosphere. As already have been mentioned just now, hydrogen is the most probable energy-rich available substrates, which is of volcanic origin. But what are the electron acceptors? I will not mention nitrate and ferric iron here, though they also could be used as electron acceptors by hydrogen-utilizing thermophilic microorganisms, because it is believed that they appeared much later in the biosphere when the oxygen atmosphere already was existing. So the most probable acceptor in the hot springs is carbon dioxide, which is of volcanic origin also, and sulfur compounds. With them it's more complicated because we don't know how much oxygen is involved in the appearance in this system. However, there is evidence that sulfates were found in buried deposits of the age 3.7 billions of years ago. That means of Archean period, and moreover, these sulfides were by isotopic composition. They are believed to be of biological origin. So the date of sulfate reduction appearance on the Earth is also might be quite early. About methanogens, so there are several genera of methanogens, of thermophilic methanogens you've just heard about, and most of them are, at least these two are living in marine hotlands, and there are not so many methanogens known for terrestrial ecosystem, at least not hypothermophilic. As far as I know, Methanothermus fervidus is still the only one, and it seems to be endemic for Iceland. We never could find it in other places. Still, the work of my co-worker Alexander Merkel shows that in moderate environments, the diversity of methanogens is much more rich than we thought it is. And moreover, this group of methanogens, this work was done by the analysis of the key enzyme of methanogenesis, MCL8 gene, and was without cultivation, just by analyzing the DNA from different hot springs. And in one of the springs, the share of archaea was the same as of bacteria, which is not usual for such low temperature. And moreover, methanogens were abundant in this hot spring, and the majority of them was presented by these two new clusters. And recently they were the first mesophilic representatives of this branch was isolated, and they were the name methanoplasmata. They are close to thermoplasma. So there is still some things to do looking for new methanogens in thermal environments. About sulfur reduction. So from the lecture of Professor Stetter, you know that there are many sulfur-reducing microorganisms, both organotrophs and lithotrophs, and among them, here I mention only lithotrophs because we are thinking about, speaking about an Archean period where there is no available organic matter. So what are lithotrophs, which can use molecular hydrogen as the electron donor and elemental sulfur as electron acceptor. Of course, these are Krenner-Hyoto, several Krenner-Hyoto groups, and there are some protobacteria and acidics. And among firmicutes, we also have isolated several microorganisms which can use molecular hydrogen and elemental sulfur and grow lithotrophically. That means to be primary producers of organic matter in terrestrial hot springs and in Archean biosphere as well. And sulfate reduction. Sulfate reduction is the feature of thermophilic firmicutes. On this tree, some groups are absent, but there are also deep branches containing thermophilic sulfate reducers, such as thermophilobacteria, and of course Archaeoglobus, Archaea isolated by Stetter's group. But there is some trick with Archaeoglobus, of which I will tell you later. Also, there is a group of Thermoplatealis which was claimed to contain sulfate reduced by several authors, but this sulfate reduction was never confirmed experimentally. And recently we succeeded in isolation first thermophilic firmicute, isolated it, and our co-workers participated in proving the ability of this microorganism to reduce sulfates. So it is the firmicute, and you see that it can really grow by utilizing sulfates and producing hydrogen sulfide, though not with an output. What is important that Archaeoglobus has all this machinery of sulfate reduction obtained from bacteria. This genetic tree is made for is a regime that features of sulfate reduction, and you see that Archaeoglobus is surrounded by bacteria, and here is firmicute cluster. And the same trees could be made for all sulfate genes of sulfate reduction, and they will up the same. And Oparin-2014 has typical crenarcheal set of sulfate reduction gene, but it also has this gene with one protein pion OA, which is absent in other crenarcheoles. So maybe why this organism can perform sulfate reduction, which could be tested experimentally both in growth experiments and with radioisotopes. We tested collection cultures of other crenarcheoles also with radioisotopes, and we got negative results. Now I want to see, I saved a lot of place here for new processes, and I want to tell about new processes of energy conservation, which we could find in terrestrial and submarine hydrothermal microbial communities, and first of them is the disproportionation of elemental sulfur and other sulfur compounds. This reaction was found for mesophilic microorganisms somewhere is late 70s or beginning of 80s by Bach in Scipionka, and it is a very strange and exciting reaction where elemental sulfur or sulfide or thiosulfate are used both as electron donor and electron acceptor for growth. So one molecules are oxidized, other are reduced. But I don't know why, but nobody has ever looked for such microorganisms in thermal environments, though elemental sulfur is absolutely available and dominating sulfur species in these environments. So my colleagues, Alexander and Galina Slavokin, they have isolated first sulfur disproportionating microorganism. You see that energy yield of this reactions is quite good, and this was thermocyclic dismutants isolated from hydrothermal fields of low spreading center in Pacific Ocean. So this microorganism can grow up to 92 degrees centigrade with optimum around 74, and it grows by reducing sulfate and sulfide from sulfur on completely medium without organic substrates, and CO2 is the only source of carbon for it. So it's little autotrophic, new little autotrophic type of life for thermal environments. Phylogenetically, it makes a new species in thermodesmutobacteria group, and it is surrounded by sulfate reducers. But we couldn't prove that this organism can grow by sulfate reduction, at least in our hands. After that, Alexander Galina test isolated other sulfur disproportionating bacteria belonging to Decaproteobacteria. One of them was found in low spreading center as well, but it is moderate thermophile in comparison with the previous one. Another was isolated, another genus was isolated from Ozon-Kazari Kamchatka, so from terrestrial hot springs. And both these organisms are also autotrophs. So that means that this new group of thermophiles growing by mutation of disproportionation, this dismutation, and also sometimes it's called inorganic fermentation. So this group is widely spread in different types of thermal environments. It is phylogenetically diverse, and it also would play a role of primary producer in terrestrial hot springs or in submarine hot springs, which fuels the ecosystem. We sequenced in collaboration with the Bioengineering Center in Moscow, we sequenced the genome of the first microorganisms with this type of metabolism. We found that it has all the enzymes necessary for autotrophic fixation, via reductive still-coated pathway, and it has all the enzymes for hydrogen metabolism, because I forgot to tell that this organism can also grow by hydrogen oxidation in presence of thiosulfate as a reference. And strangely enough, we found all the genes necessary for sulfate reduction, though we couldn't prove it experimentally. So now our next goal is to find the genetic markers of sulfur disproportionation, so that we could study the environmental relevance of this process in different types of hydrothermal systems by molecular methods. Another two processes I would like to tell you about are connected with C1 compounds that serve as electron donors, as energy substrates in hydrothermal environments. One of them is CO, which is common component of volcanic gases, both in hot springs, terrestrial hot springs, and in submarine hot vents. And Tatiana Sokolova and co-workers in strategic genetic works, and these girls are Tatiana's students. They have isolated many microorganisms of this group, although the first one was isolated by Vitaly Slikichny in 1991 in the laboratory of Zavarza. And afterwards, Tatiana and her co-workers have found that diverse firmicutes can perform this reaction, growing in 100% of CO. Lithotropically, some of them are heterotrophs, and genetically they represent very different lineages in firmicutes phylum, often on class or order level, so they are really phylogenetically quite diverse. But also we found that this feature is the compatibility of some archaea of the genus Thermococcus, hyperthermophiles. And here is the growth curve of one of these organisms. You see that CO is consumed, molecular hydrogen is produced, and cells are growing up to quite high level. And this reaction really seems very crazy because high energy valuable substrate is thrown away, but still it is how it is. When we, all these organisms grow at 100% of CO. When we decrease the percent of CO in the dead space, we found another hyperthermophile belonging to the genus Thermophil, which can also grow by this reaction. And at least at lower concentrations of CO, thermophilic bacteria of genus Dictyoglomus, this is also very big lineage of phylum level, phylum Dictyoglomi, and also contains extreme thermophiles. They can also grow by this reaction. So that means that this mechanism of energy production is widely spread among both archaea and bacteria. What is the genes? What are the genes that are involved in this mechanism? It is quite simple because it includes CO dehydrogenase and so-called energy converting hydrogenase consisting of cellular subunits and pyridexine. It is the organization of this cluster in different bacteria. Here is Carboxymethermal hydrogenophormus, the first organism of this group, and its genome was sequenced first. But then we found that Thermococcus have another organization of this group, and only one of Firmicutes also has the same organization, and this organism originated from deep sea hot vents. So we thought that maybe there is terrestrial and submarine clusters. But then when we sequenced Thermophilum Facilitropus, we found that it has the same organization of genes as Thermococcus. So bacterial and archaeal clusters, with one exception. And then Alexander Petinski, our colleague, found that there is another cluster which would be found in different thermococcal genomes. It contains also this energy converting hydrogenase, but not CO dehydrogenase, but formylase dehydrogenase. First, he found it in Korean Thermococcus strain, Thermococcus congeners, and that's how we started this collaboration with the Korean group on this new process. Formation of hydrogen and CO2 from formate was always considered a reaction with too low energy yield to be a catabolic process. But in our experimental conditions, the real energy yield was high enough to support microbial growth. And indeed, we found that many Thermococcus strains, all of them originating from deep sea hot vents, they can grow on formate, producing molecular hydrogen as the growth products up to comparatively high cell yields. Why this process can also be considered as one that might support microbial growth in ancient ecosystems. This is because formate can be produced abiotically in serpentinization reaction, a reaction of minerals with water. Not only formate, but CO as well, and methanol, and methane, and even hydrocarbons. So we can reconstruct a microbial ecosystem which starts from utilization with raw molecular weight products of cell utilization, and then producing such resolutions rate of oxygen for other microorganisms. So this is the scheme in this final variant. You can see that not only molecular hydrogen, but also CO, elemental sulfur and other sulfur compounds and formate would be used as energy substrates by thermophilic microorganisms with the other autotrophs which produce organic matter which can be used later by other members of the community. And here I want to show our experiments of about 15 years ago when we were using labeled compounds to find if there are any other processes we do not know in our laboratory cultures, and we succeeded in it. You see that we used labeled C14 labeled bicarbonate and acetate, and then we measured radioactivity of products formed, and of course there were abiotic controls, and the reaction was stopped after a certain time of incubation in situ in hot springs. So in Kamchatka we have chosen 3 hot springs with different pH, neutral, acidic, and alkaline. And in these hot springs we found points with different temperatures, 65, 70, and 85. And we measured the rates of lithotropic methanogenesis, acetoplastic methanogenesis, and what was most interesting, yes, this is the right picture. What was most interesting, we found that there is production of acetate from CO2, acetogenesis. And if there are some organisms which can do it at 65 and 70 degrees, nobody so far isolated. Yes, there is. Yes, there is Archaeopteryx fulvidus that can grow as acetogen in submarine hot beds, but not in terrestrial hot springs. And nobody knows acidity thermophiles and also thermophile which can grow by acetogenesis. And so acetogenesis would be added here as another possible process that can take place in autonomic microbial communities, which is devoid of sunlight, of organic matter, of oxygen, so no connections with modern atmosphere. And we know that acetate is a very common compound present in hot springs. And another interesting thing is acetate utilization. We labeled acetate and looked for radioactivity of CO2 produced. And we see that this process was very fast. You please know that there is logarithmic scale. So at all temperatures, at all pH, the only exception was high temperature and high pH. And such microorganisms are not known for terrestrial hot springs. We know that in deep sea hot vents there is wonderful microorganisms named geoglobus, which can use acetate, coupled with utilization of acetate is coupled with iron reduction, but we don't know such microorganisms in terrestrial hot springs. So if acetate is the key intermediate of anaerobic destruction of fermentation, and it is the bottleneck of the carbon cycle because acetate is not fermentable substrates, and its utilization demands the presence of electron acceptors for anaerobic respiration. So from these experiments we see that the carbon cycle in these communities could be closed by microorganisms which oxidized acetate as well. So I would waste the time on conclusions. I think I will make this clear enough. And looking at this old data, I think they are very interesting, and I will return with, this isotopic data, return them again and again. We published them in quite modest journal, Russian journal, Microbial, but maybe we should continue this work. Yes, I don't, there was a picture that I didn't mention, my colleague Nikolai Kliman, who did the radioisotopic work here in Moscow, but in fact it was me who said this radioisotopic in situ experiments, that it was in the trip of 1997 with Karl Stetter. Here is Eisenstetter and myself, and this was most exciting, most dramatic expedition in my life. Yes, and it's nice to remember it again. And many thanks to my collaborators, to people who helped us to organize expeditions. Gennady Karpov from Institute of Volcanology, and Godefroy from Italy, France, Andrew Reisenbach from the University of Portland, and our colleagues from the Bioengineering Center who helped us with sequencing and annotation, and our career colleagues with whom we studied the mathematization by art here. And thank you very much for your